visit a 3-day meeting that we have enjoyed, thanks basically to the extraordinary generosity and the drive of very young people, very young students. I have to underline that, specifically Berenice Jimenez and Don Juan Escalona, whom together with the extraordinary support of Federico Sanchez have done something that in many ways it's an epoch defining meeting for our university. The demography of Mexico is a young one, you all know that. And it's especially surprising to see the extraordinary depth of many of the questions that mostly the students have raised. I have to say that although I was skipped many times during the question sessions by Rosie Redfield, I'm very grateful that she did so, because I completely agree that the students should have the opportunity to ask questions. And the outcome of all the wonderful lectures that we have witnessed is this dialogue between the speaker and the young people with some naive but extremely interesting questions that reveal that we should feel very proud at the university, at the national, at U.N.A.M, at our university, that we have such extraordinary component of future scientists that by now are basically our colleagues, even if they are not, if they don't have already the title or the paper that calls them PhD or doctor or master in science or whatever. I think that really is the best way to achieve, to overcome the difficulties that all nations, all universities are facing nowadays. It really in many ways reopens our optimism in a way that reminds me of the famous phrase by Gramsci, the Italian theoretical politician, the great theoretician, about the faith in our willingness to learn and to do things. I've been asked just now to actually introduce Eros et Mazzari to you, and I accepted right away with great pleasure, because I actually became acquainted with Eros long before I had the chance to meet him, which I think is the best way to become close to someone intellectually. Sometimes when you read the book or a poem or listen to a song and then you meet the author, the writer, the poet, it can be very disappointing. This was not the case. This was certainly not the case. And I have to say that when I first learned about Eros et Mazzari, it was thanks to my very dear late friend, Lynn Margulis, who said to me once during a conversation we had, the issue of the origin of the undulipodia, the so-called misnamed eukaryotic flagella, has been solved by a young kid from Hungary. And I said, a young kid from Hungary? And she told me, well, I'm not sure how young he is, but he looks very young. He's short and has big eyeglasses. And then I finally met Eros, but not before I had spoken, or I had actually learned about him from another big name in biology. There was John Maynard-Smith. I visited John Maynard-Smith in Brighton, I remember once. He was quite a character. He was an extraordinary, by all accounts, an extraordinary scientist. And he spoke very highly about Eros again. And he said to me, you have two things in common with him. One of them is your common interest in the origins of life, which is a problem you will never be able to solve. And he was quite right on that. And the other point is that both of you are theoretical biologists. And indeed, there is a brotherhood or sisterhood, to satisfy Rosie Redfield. There is a certain whatever-hood of those of us that like to think that biology has to become a very strong combination of empirical observations, of experiments, of understanding, watching, looking at the actual organism that has to be put into the frame of evolutionary theory with the broad understanding or the broad definition of what evolution is today, but we cannot do without theoretical biology. And I truly feel here a sense of brotherhood with with Eos, because if you look at his publications, most of them have actually to do with very deep analysis of a number of issues that are related to the origin of the genetic code, the origin of coenzymes, the reconstruction of or analysis of the RNA world, and a number of things that actually provide not only theoretical or mathematical descriptions of a number of phenomena, but also made specific predictions about what we should see in experiments, what we should see in nature, and so on. And this is a very rare achievement because most of the people that do theoretical thinking in biology actually do not go into that direction. When I finally met Eos, I was quite impressed by the easiness of his conversation, by the natural way in which we got along together. It's, with Eros, I can say that I feel that I have a friend on the other side of the world living in Hungary. And one additional thing that we rapidly discovered is that there was a time in which both he and I were ruled by the same emperor, the Habsburg emperors. So perhaps some of this polite way, multicultural attitude that he has is very easy for us Mexicans to understand in that sense. I would like to end with a final issue, namely that a number of times we have shared private jokes. Remember those about our colleague who you define as a witch using a broom that she flew in our room. We were in Crete. And then I say, yeah, she must be a frequent flyer. I'm not going to say the name of the lady, who's a very attractive, not so intelligent colleague, but very, very good to talk to. And so on. But finally, there is a question that troubles a number of people. Exactly what does Eos Esmatzari means? And according to Lynn Margulis, you may say if this is true or not, Eros is the Hungarian for Eros, the god of love, no? And Esmatzari for Saint Mary. No, okay. So Lynn Margulis was correct in a number of things, but not in this particular case. In closing, let me describe to you a brief conversation that Eros and I had once. We were describing the behavior of the Hungarian nobility. And Eros told me that it was very common during the Middle Ages, even up to the early 19th century, to have all the members, the male members of the Hungarian nobility coming together to a place, hiring good chefs with good food, with good wine, and then starting a party that will go on for several days until they were completely drunk, and they decided, and they will normally burn down the place. I think that we have seen flames arising during these discussions, so I think it's good to remember that traditional Hungarian trait to see how his discussion today, his lecture today, will put your brains and ours aflame. Eros, welcome.
Dear Professor Lascano, thank you very much for this introduction. And I apologize, I know that I look like Jason Robards in Once Upon a Time in the West entering that inn. You remember, it's a fame. I don't have manacles, but it's, you know, the temperature that I have to worry about a little bit. Yeah, after all these nauseating references to our work, there is an uphill battle here for me, I am afraid, to meet all these expectations, but let's see. First of all, that's a reminder that I deliberately put in the beginning. First of all, because, you know, age is coming, and I tend to forget about things like that. The second thing is I put it in the beginning because should I fall asleep during the lecture, then definitely I have said it, and then the most important part is there. Third one is that somebody told me during the meeting that blue light is good, you know, against the jet lag, so all the slides will be in blue. One more thing, and this we should clear in the beginning. It has been a long meeting, and it's the very end, and we are slightly tired. So I would like to ask you a question. So if you wish, I can finish the talk exactly in the 45th minute, if you wish. I might go on, that's another alternative, for another 10 minutes or so. So I would like to explicitly ask the question, so please put up your hands if you allow me to go 10 minutes beyond the 45 minutes. Thank you very much. No, I'm really worried about these things, so thank you very much for this kindness. And I want to emphasize also, as many other speakers did, that I'm extremely overwhelmed by all the help that the students have given us. And the students remind me of the students in Hungary, but 20 years ago, right? They are nothing like you today. They have deteriorated a lot. I hope you are not going to do that. So thank you very much indeed. It has been an absolute pleasure. Now, with these words, we would like to get down into business. You know this slightly petrified gentleman up there. Actually, he was a very sweet guy, not cast in stone in his personal behavior. And he actually put up a theory of evolution about which Daniel Dennett says Darwin's dangerous idea. Actually, he should have said Darwin's infuriating idea, because especially in the social sciences, people are so pissed off, right, that you can summarize the essence of the theory in one slide, and it has got an amazing explanatory power. Many people in social sciences believe that complex phenomena need complex explanations, preferably not testable, right? Because I was at a place where I was mixing with social, I know what I'm talking about. But indeed, if you read Darwin, this idea of him that these things that are summarized like this with Maynard Smith, these criteria that you have these units, and some hereditary traits must affect survival and/or fertility. And if you have units like that and a population of such units, evolution by natural selection can take place. There are also certain quantitative conditions. But that's only one page, and this actually Darwin realized quite long before the publication of The Origin of Species. Mind you, in the present formulation, in this formulation of Maynard Smith, there is no reference to genes or organisms. Why? Because evolution is a dynamical process, and if a unit, wherever they are, whatever level, they satisfy these conditions, a kind of Darwinian evolutionary behavior is likely to be observed. So that's a strength. That's the most general statement. Also, and then I finish this introductory laudatio of the Darwinian view. When you read Roger Penrose, The Emperor's New Mind, which actually you should, then he lists a number of super theories regarding the explanatory power they have, and there are only 3 in that book. One is quantum field theory, the second is general relativity, and the third one is the Darwinian theory of evolution. So we are in good shape if we follow this path. Now, this basic dynamic, surprisingly perhaps, has led into what now we call the major transitions in evolution. This is a popular book, the Spanish translation of it, and in the original book, and also in this one, you can find these transitions. I don't want to go through the table. This is actually that there are 8 steps in evolution, and there are 2 important things about it. These are not the 10 commandments. First of all, because it's 8 and not 10. Secondly, it was an abstraction away from what we had at that time. This is not cast in stone. This, is as an empirical generalization. And as you will see shortly, I actually have modified this. But one more remark here. This is again the same list from the original book, and you see that there is an asterisk associated with certain transitions, and those transitions traditionally are regarded as difficult by people in the field, and also, interestingly, they tend to be the unique transitions. What do I mean? You know, you cannot be sure that there wasn't quite a different kind of attempt at a genetic code, because everybody has gone extinct who did something else. So it's just operationally unique in the sense that there is a monophyletic ancestry of what we see today. Otherwise, you don't know, because the things may have been lost in deep time. Now, this is exactly what I would like to say, that I have revised and partly reconceptualized what we have done, and is due to come up early April in PNAS. Right now it's under embargo, but so soon it will be available, and it summarizes what actually it's slightly less and slightly more what I'm going to talk about today. Now, as you know from the original book, these were the recurrent things in the transitions that you see quite often. Not exactly with the same weight for all the transitions, but they are recurrent to such an extent that they really merit some mention as overarching themes. You know, one is this individuality transition type of thing that Rick and Stu were also talking about. Then there is a very important thing, the division of labor / combination of functions. Then the origin of novel inheritance systems. You see, there was a lot below and before DNA, and above and after DNA. You know, you have things like the RNA world. Then you have epigenetic inheritance, and you have symbolic communication. Because what you heard in the previous talk, with its digital nature, the open-endedness, infinite possibilities with finite means, that describes contemporary genetics as well as language, right? But language is at a higher level, and evolves partly differently, and all these give rise an increase in complexity. And there are two other corollaries here. One is that often there is no way back, because things get entrenched. You make a transition, and then things get accumulate, and then it's very difficult to go back. Sometimes you can, but in many cases you can't. And central control usually is something that helps in the solidifying of the new level. But this is from the previous books. Now, David Queller, when he wrote the book review in '97, all the book reviews were extremely favorable, and we were very happy with that. But in this book review, actually developed the theory further, and this is very important. He realized that there are two types of transition that we were dealing with. One is the fraternal one, and Stu was and also Rick was mainly addressing this. But there is the egalitarian type of transitions, like, for example, the origin of protocells from unlinked replicators, or indeed the origin of the eukaryotic cell. The most important thing is that the guys who come together in those transitions are not related to begin with, right? So they have to come together somehow, and you have to resolve the conflict in ways that are different from the conflict resolution in the other parts. And there are, initial hurdles. In the egalitarian transitions is the control of conflicts, and in the fraternal transitions is the initial advantage, because it's not immediately obvious if everybody is the same. So what is the advantage of being together? Because there is no complementation of function at that level. Yeah, I can use this. Yes. So now we should start a little bit considering some details of the transitions, and let's start at the beginning. I'm going to use the era about early replicators and protocells as a kind of case study, because they show in a very simple form many things that came later, but these are easier to handle. So I will spend some time on the transitions along, protocells, origin of chromosomes, and these kinds of things. So obviously what you would like to have somehow to launch the whole system is a chemical instantiation of a hereditary replicator. Not just replicator, which is a simple autocatalyst, but actually which has many states, and those states can be propagated, just as the conditions for the evolutionary units. You remember the slide with Darwin. Now, I have to disappoint you. The origin of hereditary replicators with a considerable hereditary potential is an unsolved problem, right? There are some beautiful partial results. One of them is by Günther von Kidrowski, a brilliant chemist, who in 1986 has figured out that if he designs a hexadeoxynucleotide analog in that way, and then if he puts into the test tube the 3 nucleotides as well, they will spontaneously anneal. This will be in proximity. That will catalyze the formation of the covalent bond. And since the guys are short, with a certain chance they are going to dissociate. But this guy is the same guy as this, is just the opposite. Both of them can start another replication cycle. No enzymes are needed in that process. That was the first artificial chemical self-replicator, and it started a zoo. Actually, if you now Google for the term replicator, you are close to finding more reference to the term replicator in chemistry than in biology. And that's actually because of this pioneering work. Since he was a nobody, it was duly rejected by nature and science alike. One of the referees said that it was in the referee comment that this paper is incredibly pompous. And the reason for this was that Günther said in the paper that this is the first example of a non-enzymatic, which was true. But the referee couldn't bear it to such an extent, right? So that's for the young students, that science and also your career is not moving like this ball, when you learn physics, with constant velocity in a straight line. It's actually very far from that. Now, the problem, as you can see from this, is that if it's longer, it's not going to separate by itself. No, you can of course heat it, but RNA is labile, so if you heat it, that's not a good idea, so you are in an impasse. The other way to do it is that you create a ribozyme, a replicase ribozyme, that would do the job for you, but of course that replicase ribozyme should be able to replicate another copy of itself, as well as other ribozymes of the same size. And we don't have this, there are attempts, we don't have this either. There is no generalized replicase ribozyme. That, although people are trying desperately, because it's regarded by some as the holy grail in the quest for the RNA world, also it might result in a Nobel Prize. So there is a race going on, but so far it hasn't been successful. So this is an unsolved problem at the moment. But from now on, I will pretend that somehow the problem was solved. We don't know. In science, you never know. It might be an impasse that ultimately, calls for some radical novelty in our thinking. But let's see how it goes. Before we continue, I have to quote this. This is from my intellectual grandfather, G.B.S. Haldane, because he was the intellectual father of John Maynard Smith, a formidably clever man. The Nobel Prize winner Peter Medawar said about him that he was the cleverest person he has ever met on the earth. And indeed, he was intimidating, and this is a very, very important thing. He was not only one of the founding fathers of population genetics, but also he made work in enzyme kinetics, the origin of life, and he was probably the best science popularizer at the time. So it's good to read it aloud. An ounce of algebra is worth a ton of verbal argument. This is very important because there is a little bit too much of this in certain areas of evolution biology, whereas people should try. It's not always possible, and in many cases it's not easy, but they should try to put it down to some model where they can actually make the assumptions really explicit. Making a model forces you to make the assumptions. There cannot be anything hidden, because then you cannot do the associated computer program. So I took this quite seriously. I occasionally fail because the thing is too complicated. So the dynamics is extremely important in order to tackle the evolution processes. And you know, for example, the origin of life, and this is very important. The origin of life, as well as all the other transitions, is as much about the dynamics of the process. You must get to working machineries as much as structures, because dynamics produces structures, and the emerging structures give you constraints for the further dynamics. So therefore, you know, if you want to do proper science about this, ultimately this has to be the goal. Now, I present you with an example. As I said, now I'm assuming that the replication problem has been solved, that there is still an obstacle here, which was realized by Manfred Eigen in 1971. This is actually a very important moment. He is still alive, a Nobel Prize-winning biophysical chemist, who in 1971 published a landmark paper, Self-Organization of Matter and the Evolution of Biological Macromolecules. It's a bloody long paper in the Naturwissenschaften, but in English. You can download it freely somewhere from the internet. It's marvelous to read it even today. It's crystal clear. So what he discovered was the following. If you have an RNA or whatever, a nucleic acid replicator, and it's digital, and there is a copying fidelity, which is, of course, the inverse of the mutation rate, what happens is that if let us suppose that this is the wild type, you know, here, and then come the erroneous copies, you know, that's the Hamming distance, how far you are from the master sequence. What he discovered was that as you increase the error rate, there is a point where you actually lose the genetic information. It will be in the noise somewhere here. That's the error threshold of replication. Now, of course, population geneticists knew the mutational load, but they did not realize that the mutation rate also constrains the selectively maintainable genome size. That needed a fresh mind. So he pointed out that there is a problem. You know, we already think that the replication problem has been solved, but now there is another one. You know, it's one of those dances, you know, with the 7 whales, but in this case, the lady under the whales is becoming uglier and uglier, you know, with every whale, because, you know, there is a further problem. So Manfred Eigen realized that early replication must have been error-prone. If early replication was error-prone, you cannot maintain a long genome, right? Then comes the idea, okay, you shouldn't maintain it in one piece. You take a pair of scissors and you cut it into pieces. Yes, but you know, these molecules will have different sequences, different shapes, different phenotypes, so the replication rates will be different, and therefore they are going to compete against each other, and that will be the end of the genome. This is, my dear evolution biologists, one of the earliest and clearest realization what nowadays we call intragenomic conflict, right? So that was again a very important observation by Manfred Eigen. Now, what can we do? Now he proposed a solution to the problem, and we are already in the field of cooperation, right? He wanted to solve the problem by cooperation, rightly so. The concrete suggestion is wrong, as we shall see shortly, but Okay, so there are these replicators, each one producing a replication cycle. So they are autocatalytic, but there is also the heterocatalytic aid, and then it helps, the replication of the other member. That's the famous hypercycle presented in 1971. And this is indeed ecologically stable, right? It's either goes into a fixed point where, everybody has a constant relative concentration, or if there are many members, you get what we call a limit cycle. That's also a stable oscillation. Beautiful. Then when Meinhard read a book which was later published by Eigen and Schuster, he said, wait a minute, I don't understand this. Yes, because there are two little problems. Let us take this only one. So here is the hypercycle, and here is a parasite, right? Here is a parasite, and the parasite accepts the help of this guy, doesn't help the system at all. Now, as they say in nasty physics textbooks, it's an exercise to the reader to prove that if this arrow is stronger than this one, no matter what else happens in the system, but the parasite is going to finish off with the cycle, right? Again, the genome has been lost. So what? What can we do now? Obviously, people found out that somehow it would help to put the whole thing into compartments. Yeah, what? To create new higher levels of selection, because there is no higher level here. There is an ecological system with certain interactions, but it doesn't behave as a unit of selection. Okay. And this is, again, I said this is a didactically good example because analogous problems arise at all levels. As Stu said, again and again, we have to face the same problem when we want to arrive at higher levels of evolutionary units. So let's look a little bit about the compartmentation issue, because that's, again, something that is important. Let me jump to that. So this is one of the interesting investigations. Imagine that you have a metabolic replicator system. What does it mean? That here you have got replicators. These replicators catalyze a common metabolism, individual steps, and they can feed on the product of the metabolism. So they catalyze the metabolism, and they take out their share that they are eat so that they can replicate. Now, amazingly, this system, you wouldn't believe it at first sight, is not ecologically stable, right? Because the fastest replicator is going to overgrow the others, and the metabolism, since all the members are needed, is going to go down the drain, so the whole system is lost even without parasitism. So we were interested in the year 2000 or something. What happens if you put this system into a spatial setting? What is the spatial setting? It's a passive, a poor man's form of compartmentation. So, no, come on. It's a poor man's form of compartmentation, which means that you are on a mineral surface, or you are in porous rocks, in little holes of rocks. So that is the environment in which the replicators are living. By the way, such surfaces must have been rather important during the early evolution of such processes from the chemical point of view also, but I'm focusing now on the evolution point of view. So this is an unstable system, yet if you model it as a system that is spreading on surfaces, and that you can do by a so-called cellular automaton, it's a technique. I don't want to go into details because it will be a waste of time in a way. It just means that in each cell you have a molecule or not, and there are two types of neighborhoods. The replication neighborhood, you need a template so that you can put a new copy into an empty site. And you also need a metabolic neighborhood, which means that in a finite neighborhood, all other members of the system must be there, because otherwise there is no local metabolism. and there is diffusion into 2 dimensions of the system. It's very technical, but this is exactly the point. If you don't do this, you have absolutely no idea what is going to happen. It's extremely important to do these type of models, because otherwise it's endless verbal argument. Now, what we found, amazingly, was that this system is stable in a spatially explicit setting, and it's stable as a system, as it is here, and also is stable against parasites. The parasites appear, but they cannot take over. How come that this simple trick of putting these things onto a surface solves so many problems at once? The reason is very interesting, because there is a cost of commonness and an advantage of rarity in the system. So if I am a replicator, for example this one, I replicate faster than the others, what will that mean? That will mean that very frequently you will find yourself in a situation where you look around and it is just other copies of you. But that patch will be metabolically inert because you are missing the complementary partners, right? Whereas if you are a slow replicator, if you look around, usually you will find that it's other types of replicators around, and it's jolly good to have other replicators around because they are the complementary partners in the local metabolism, right? So this system is extremely stable. You can ask a following question if you are a trained population geneticist. Okay, what about the diffusion rate? The diffusion rate amazingly doesn't matter. It can be arbitrarily high. Why? Because this system then goes over into a system like this one, right? If the diffusion rate is very high, then this approximates a system which looks like this. There is a random pool. These are the local patches. These are the local introductions. And because the diffusion rate is high, then there is a harvest, and the harvest can go anywhere into the local patches. Rick Michaud was the first, according to my knowledge, I think in 1983, who applied this kind of model to these questions of early replicators. And where we went further is that we demonstrated that this otherwise competitive system, the metabolic replicator system, if you are putting it into a spatial context, is stable, it's ecologically stable, it's also mutationally stable against the parasites. Now, here is another system that we are going to compare it with. That's about the origin of efficient replication. So I am now forgetting about the metabolism, and I'm concentrating about the following question: how do efficient replicases arise? As I told you, we don't have such a thing yet, but I assume that the chemical problem can be solved, but I want to solve the dynamical problem now, right? So here is a replicase RNA, and here are other RNA molecules that don't do the work, but they can accept the help. Now, the tough thing to model is, for the system I mean, is to model it as a parasite. So, which means that this arrow will be thicker than this one, which means that you have a one-membered hypercycle. You know, it's a template replication, but it's also a replicase enzyme helping, you know, also enzymatically the template replication. What happens? Again, if you are in a well-stirred system, the whole system collapses, the parasite takes over, parasites take over, goodbye, you can forget about it. Now put it onto the same type of environment, a setting, you know, that we just discussed, and here are the results. What you get is the following. We assumed, by the way, that there are trade-offs between different important features of the replicators. So if you are a very accurate enzyme, then it means that you are not very fast, right? If you are fast, you are not very accurate. If you are short, then you would be fast as a guy who accepts the help of others, but you are not going to act as an enzyme. So it is as hard as possible for the system. again, you know, if you take a, you know, some kind of a chemical thing and liquid water and you stir it, you know, everything collapses. But if you are on surface, that is what happens, right? So in the simulations, this is the timeline. The error rate of the replicases decrease, that's the average. The replicase activity, how efficient you are as a replicase, you know, putting one nucleotide, increases. The length of the molecules also increases, just because you are making the dynamics of the surface. And here is the stationary population. There will be parasites that are shorter, but look, there is no way that the parasites can kill the system. And of course, there is a partly similar argument as before. However, there is a big difference. If you increase the diffusion rate, that system collapses. Now how? Now this is again why, you know, to make models is important. Why? There is one crucial difference between the two systems. In the metabolic replicator system, if you are a ribozyme catalyzing metabolism, then one copy of yourself is enough to exert your contribution towards metabolism. Yeah? One copy of enzyme can catalyze a reaction among small molecules. One replicase molecule cannot catalyze its own replication because there is no template, right? You need at least two. And that single little problem makes the system vulnerable to high diffusion rates. If the diffusion rate is small, then the system survives. It depends, put it in another way, whether molecules, as kind of altruists, can scratch their own back or the back only of others. That's a big dynamical difference in these systems. So the moral is that spatial structures on mineral surfaces is good for you, must be good for you, because this is what the theory predicts. Okay, but as I said, this spreading on the surfaces is a poor man's form of compartmentation. Why? For example, thank you. I will look like, you know, a soldier with many weapons. Can you switch it on?
Let me take this off because otherwise you will have interference.
Yeah, exactly. Okay, thank you very much. So we continue from here. Sorry for the interlude. It's not due to me. So why is the surface-bound system limited? Partly because in a small neighborhood you can have only a few different enzymes because there is no space for it, right? So the number of replicators that you can maintain, I mean the different ones, is limited by the size of the metabolic neighborhood on the surface. So you can actually maintain systems, but they must be small. So the efficient systems are this one. So here you have a bag of genes, but those genes are like the metabolic replicator model, so they have a synergistic effect on the dynamics of the system. The different types are important for the metabolism within the protocells to run efficiently, and there is an optimal ratio of the two guys, because those both have to contribute. Mind you, this is actually a difference to the usual altruistic model, where the altruist does some work and the other doesn't. Here, you have several types of altruists that have to act together synergistically, and they are unrelated to begin with, right? It's a completely different kind of game that we saw previously. Now, what happens in this case is the following, that inside every compartment, there is a tendency of the fastest replicator to take over. You know, we don't cheat. Competition is there. Now, however, what you have is you have these stochastic effects. When you divide, every molecule can decide whether to go left or right, and that generates variation at the level of the protocells. So there will be systems that have a better internal composition. Other systems will have a worse internal composition. So they are going to compete with each other. And despite the fact that there is internal competition, selection that is acting among the cells can stabilize the population. And this is fully analytically solvable. The two-level, this is a multi-level selection, right? But one of the earliest models, and in fact, it's a model that treats both selections at exactly the same resolutions, and the two are coupled, so one follows from the other. And this is actually the mathematically sound version. This is somewhat amateurish mathematically, the first version of it, but nevertheless it has been proven that it's completely rigorous. So this kind of interaction to put things into protocells is actually a very good idea, because protocells give you an efficient form of compartmentation, much better than the poor man's form of population structure. So you are at the protocell level. Yes, so, but, you know, theoreticians have certain limitations. As they say, you shouldn't only talk the talk, but also walk the walk. And this is exactly what's happening right now. There is a brilliant molecular biologist, Andrew Griffith in Paris, the inventor of the in vitro compartmentation technology, and together with whom we have developed a microfluidic device. A microfluidic device that can enforce a life cycle on small droplets. Every droplet can be fed with the raw materials, and they can be forced to divide, and they can be selected according to the internal working of the agents that are inside the protocells, and those agents are replicating ribozymes, right? The first publication is going to be submitted. I mean, this is hell of a lot of work. I mean, as a theoretician, I have an incredible respect for Andrew, because not only can he solve these problems, but he actually solves them on time. Now, if you are an experimentalist, that you will appreciate what that means, to actually finish an experimental system on time, as you promised. And this is what it's absolutely awesome. So by this, we can actually test the theory in concrete molecular terms. I just wanted to mention it because this is a very important aspect of the further work. Ultimately, the soul of natural scientists is saved only by the lab, right? And so this is extremely important. Okay, now we go from this into a question which is related to another cross-cutting feature, this division of labor problem. As you know, it appears in several contexts. And now I would like to ask, again, in the context of this molecular system, because it's simple to follow. And the question I'm going to ask, what is the condition for the successful selection of specific enzymes? What does it mean? Specific enzymes mean that I catalyze a particular reaction efficiently, and I don't catalyze the others. As Henrik Karcher, the inventor of metabolic control theory, modeled in the 80s, it's natural to start with sloppy enzymes, and sloppy enzymes catalyze different reactions, but inefficiently. How you get to from sloppy enzymes that are generalist to efficient enzymes that are specialists? That's the question, and he couldn't solve that problem. We had a go at it, and here is the solution to the problem. It gave an unexpected insight. So again, we take this stochastic corrector model, as it is called, and we ask the question, if the black and the white and the other colors guys, that are the different enzymes catalyzing the core metabolism, if these enzymes are inefficient and generalist and specialist and efficient, so how can you make this evolutionary transition? In order to do this, you need a metabolic pathway, but there is a novelty which they didn't put into it. There are waste directions, right? Which means that reactions can go in wasteful directions that are not good for the system. And since the enzymes are not specific, they can also catalyze wasteful reactions, right? Don't assume shouldn't your point of departure be what you see today. You cannot start with that. So that's a mess to begin with. Now you continue and let evolution work. There are mutants that make them less efficient and more efficient and whatever. And you apply this system. You have to, of course, somehow model the active sites of the enzymes. I am going to skip this detail because I don't think that it's very helpful to get into it. There is some theoretical chemistry behind it, but we shouldn't bother about it. Suffice it to say, we can model, we can have a mapping from the traits of the enzymes to the reaction rates that they are catalyzing. So what happens in a system like this? So this is a characteristic run. What you see in time is that the number of waste catalytic directions goes down dramatically. The efficiency in the different directions of the system increases steadily. Consequently, the waste flux goes down. I mean, these are several orders of magnitude. And the direct flux that produces the substances that are important for the replication of the replicators increases. But notice that there is some strange curve there. What is this? Independent replicators. So the independent replicators means what you find in the stochastic corrector model. It's a bag of genes, no genetic linkage, and you cannot evolve efficient enzymes in the system. Why? We shall see immediately. Here it is. It's because of the assortment load, ladies and gentlemen. What does it mean? The assortment load is that you can have cells that don't have a single copy of a particular enzyme type. And by the assumption of the model, that cell is inviable if the enzymes are completely specific, right? So this guy is inviable because there is no green enzyme there. However, if the enzymes are not specific, look at it. It doesn't matter if there is only a single enzyme there, because you see, with the color, it shows that it can catalyze both reactions. It's not very efficient, but it's better to be inefficient than dead, right? So there are jokes about this, right? So the stochastic corrector model, unless the chemical reactions are orthogonal because of a chemical point of view, unless they are orthogonal, and then you can be specific. If they are not orthogonal, you are going to have generalist but sloppy enzymes. And what's the condition to leave this impasse? The invention of chromosomes. Because chromosomes ensure, if there is a segregation mechanism, that both offspring cells are going to inherit the synergistic partners, right? So it's surprising. We didn't know this. So it's a very important driving force towards the origin of chromosomes in order to have specific enzymes. And then, of course, the protocells are going to be incredibly efficient. Now, where are we? Let me step over this now. You are not going to see this, I believe, but equipped with all this knowledge, I have, in this particular paper that I was alluding to, I have revised the whole table of the major transitions. And let me explain this table. You don't have to see it. What is important, I'm going to explain by pointing. So here is the origin of blah, 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 so the different stages. Here is the formation, maintenance, and transformation phases. This comes from Andrew Burke, who said that these transitions should conveniently be cut into different phases. And, these are always, there are 3 phases associated with everything. Then there are the relevant transitions in individuality. Here, the new type of information storage, use, and transmission. And there is also an associated category, the limited transitions. Now, there are a few important changes. One of the important changes, look here, the origin of plastids is an independent transition. That is because, of course, you can be a completely functional eukaryotic cell without the plastids, but the acquisition of plastids opened up an incredible radiation, which also has all these characteristics that you see there, right? The transition, individual, and so on. So now, they were separated in time by almost half a billion years, so from the rest of the eukaryotic cell. So that's now an independent transition. What you don't find here as a separate, so that was a promotion. There was also demotion, step downwards. And for example, you don't see the origin of eukaryotic sex as an independent or equal level transition anymore. It figures here, that actually twice, because it might be the case that at the very origin of the eukaryotic cell, you had some primitive form of sex. Why? Because certain archaea have this type of thing. Certain archaea can fuse, can have genetic exchange, and then they can separate from each other. So that, talking about the origin of sex, that's a character that you could actually start with, already at the very, very beginning of the eukaryotic cell, and is the ritualized form of this, in the form of contemporary meiosis and mitosis that follows probably later. So that's one of the important changes. And mind you, as you saw from the previous lectures, it is absolutely not clear now what followed what. Phagocytosis, meiosis, mitochondrion acquisition, mitosis. Meiosis could be more ancient than mitosis. One of the hints is that mitosis is extremely variable. Meiosis is extremely standardized, right? So there are all kinds of things. I don't have the time to go into it, but there is a reason behind all the changes that I have made. Now let me also mention something, which is the limited transitions. The limited transitions mean the following thing. The limited transitions is that you do something which has the smell of a major transition, but it did not have so far a major phylogenetic impact, right? So you can have a 21st and a 22nd amino acid in certain organisms, or for example, in the case of the ciliates, you have a within cell soma and germ differentiation. These are analogous cases to things that happened in other major transitions, but so far they had a limited impact. These are the associated limited transitions, but they can be, of course, very important because you can study it in a micro mode, so to speak. So that's also one of the novelties. Also, that's also important. Look, that two previous transitions. That the naked genes escape into compartments and the chromosomes form. Now they are these phases of a single transition, which is the origin of protocells. By this, actually, incidentally, the table is not so top-heavy anymore. There were too many transitions at the beginning, and I didn't like that. I think that now it's much more even, right? So the resolution is, I believe now, much more even across the table. So, but you can study then this, in 1 or 2 weeks. Okay, so there are 2 other types of transitions, not in the table, but I discussed them. There are 2 others, the recursive transitions. Now, what does it mean? When analogous transitions happen from the product of the previous such transition, I will give you examples in a minute. And this is what interests me much more than anything else before. That is what I call the filial transitions, when you have an origin of a new Darwinian system, but between two already existing levels. And we shall see them in a moment. So this is a case of the origin of plastids. In the origin of plastids, you have repeated and recursive transitions. It's just not, once and goodbye. There are, of course, primary endosymbiosis, that's fine, but, what happens afterwards is that you can, for example, phagocytose at the different parts. Here is a secondary endosymbiosis, and in a secondary endosymbiosis, there is a eukaryotic cell that eats another eukaryotic cell that is photosynthetic, right? And this is the acquisition of a plastid that is of eukaryotic origin. You make the same thing, but you repeat it, right? What you did before, and then you get. It's like these two crazy Russian dolls, right? You open it and there is another doll and there is another doll. Here are the tertiary symbiosis, right? When you do it for the third time. And there are interesting details here, I don't have the time. But also, there is a repeated case now, it seems. Here is Paulinella. And Paulinella, it seems, it's a case where a cyanobacterial origin seems to be happening now independently. Mind you, I don't know of a single example which would be the case for the mitochondrion. I don't know about secondary mitochondria in the way that you would have a secondary bacterium which would do respiration for you. For example, imagine that something like Giardia. Some Giardia could have, tried again to be aerobic, and that lineage could have engulfed an aerobic bacterium and could have gone into that direction to try it again. I don't know a single example about that, but the plastids are full of these cases. So here is another one. Here is a recursive transition, but of the fraternal type, and this time about multicellularity. This is siphonophore. And this is a nasty creature. You must have seen it, but I hope from at a distance. Don't try to mix with it. It's the Portuguese man of war. Now, these guys are actually modified zooids that resemble otherwise free-living animals, right? So now the whole animal was adopted as a unit, right? Not a cell, but as a unit, which then is used with differential gene expression so that they act as organs, but it's very clear that they are adopted or co-opted organisms that, of course, are related, so it's a fraternal transition. But again, you can have a repetition of a fraternal transition, but now you are using already animals to create an animal again. How am I doing this time? Did somebody watch where are we? How much time is left? Sorry? What does that mean? How many minutes left? Nobody knows. That's actually very good news, right? But I know that time will be soon up, but there are some nice issues still. So these are the recursive transitions, and obviously they are extremely exciting, and they can happen, with cells, with multicellular organisms, egalitarian, fraternal, and many of them are much more recent. So again, they offer some extremely interesting comparative evidence for, generalizations and so on, like Sue mentioned. I mean, these have to be analyzed from the same point of view. And there's plenty of work, right? The major transition thing is basically at the beginning, I would say, and not at the end, if you consider all these things that I highlight. I jump now, and now I go to the filial transitions. what was a filial transition? A filial transition is the origin of a new Darwinian system in between two existing levels, right? So if you take multicellular organisms, there are cells, there are organisms, and here is the case of the adaptive immune system, right? I deliberately take this particular and classic figure. This is a figure that was drawn by U.N.A.M. U.N.A.M got the Nobel Prize for immunity. And look, in this he actually describes what we know about the generation of antibodies in vertebrates. So there is a bona fide Darwinian type of process whereby you are producing the antibodies, and then there is a fine-tuning of it by mutations, which is called affinity maturation. Moreover, look at the title, the generative grammar of the immune system. Why? Because there are a finite number of operations like somatic recombination, somatic hypermutation, and with these finite means, you can generate potentially a number of antibodies for which it was calculated that it covers basically the whole possible shape space. That was calculated by Alan Perelson. That's again a system which resembles its open-ended, unlimited heredity type of thing that is so important for genes, so important for language, but also happens for the adaptive immune system. Now, it's actually very interesting that the origin of the adaptive immune system is not one thing. It originated twice independently in the vertebrates, and the details are different, but the Darwinian nature is the same. We are going to submit soon a paper about the comparative analysis of these systems, right? From the point of view of transitionology. So that's one example. Now we come what I think is much more exciting, and this is where more than 50% of my work goes these days. Okay? Eva Jablonka and Marion Lamb moaned in 2006 that not only Maynard-Smith and Satmari, which is okay, but even them have forgotten about the nervous system before. Yeah? I mean, it's hardly any mention of the nervous system as such in the major transitions in evolution. And you know that there is a point, but let me quickly tell you what the point is, according to my understanding. So here is evolution of the brain, right? So if you look at the cortical structures from amphibians up to humans, you will see that the cortex became more and more complex. Let me tell you one thing, this is absolutely important. There is an urban legend. It's like the 17 words for snow in the Eskimos. But it's such an urban legend that you find it in neurobiological textbook. What is it? If you take a slice of cortex from a human and the macaque, you can't tell the difference. It's absolute bullshit. Every good neuroanatomist knows that it's not the case. But just by looking into the microscope carefully, he will be able to tell whether it's from a human or just a primate. Why? Because it's a more complex structure. The distribution of the cells is different, and there are more cell types even. Okay, now this is important, my beloved sisters and brothers in Darwin. It's really very important. There was a brilliant person, Santiago Ramon y Cajal, one of the most important neuroanatomists of all times. In his autobiography, he asked what he really wanted to understand: what is the difference in the brain that gives you the, as he said, it's very sweet all the time, the psychological nobility of Homo sapiens. And then he mentions what are the traits, including language, for example. Now, this is what I am up to And I think I have one important component where there is, I believe, a radical difference. And I know that this will be the last 3 slides or something like that. So Changeux, in 1973, who almost got the Nobel Prize for allosteric enzymes, became a neurobiologist, and he formulated this picture of the development of the nervous system. There's overproduction of neurons and synapses. You eliminate what you don't want according to functional criteria, and that's the functioning nervous system of the adult. It's a true picture, we know, but it's only a partial truth. There is a limitation. It's explicit in his writing. It's actually there. It's amazing. An organism cannot learn more than is initially present in its pre-representations. Everything, it's a cognitive homunculus, right? So there is no real novelty. You know, you make this exuberant overproduction, and then you chip away what doesn't belong there. It's like the advice of Michelangelo how to produce a statue, but please note that the connectivity in the statue as a piece of stone is maximum. It's not like this in the brain. The brain is extremely sparse. Most of the possible connections between neurons are not there because there is no space for it. If you wanted to connect all your neurons, right, which would then in a way contain everything that is possible, then the white matter, would be much bigger than your head, right, by a cubic meter or something like that. It was calculated. So there obviously is a problem here. And then let me tell, here is the final slide, not the final, but the final content slide that I want to show. The picture of the brain has radically changed empirically. Why? Because the neurobiologists, by using clever technique like this long-term 2-photon microscope in vivo, what is mean that you install a peep show in order to look into the brain of a behaving animal, right? And what do you see? You see that connections between neurons grow and they become eliminated in real time. That's a paradigm shift, but why are you doing this? It's bloody costly in terms of ATP. And if you take an off-the-shelf model of the brain, there is never a possibility of that. Most of the neurobiological modelers don't want to know about this. It's still not there. I mean, 90% of the models don't know about this phenomenon. It's absolutely amazing. Why? Because, as for example, Kierkegaard, his name is not here, but this is a review. Kierkegaard made the following model. It's a verbal model, but this has to be mathematized also. That this is the expansion renormalization model. So before learning, this is the situation. In learning acquisition, you produce new synapses. In learning consolidation, you eliminate those ones that are not necessary for maintaining the long-term memory what you have acquired. So this Changeux type of picture of overproduction and pruning becomes an iterated game. It continues in your brain now, right? It's real time. There is a distribution of synapses in time. I don't want to go into the details, but Kierkegaard says, look, it seems that there is a Darwinian-like process in the brain. There is no model of it, but there is an observation. So what I would like to know, and this is what I am working on together with this guy, Crisanta Fernando, what would be a design for an evolutionary brain? How can you implement bona fide evolution in the brain, by what mechanism? And I'm happy to say that in contrast to these toyish models that we produced before, I think that 3 weeks ago I realized how to do it. So obviously that's my major thrust at the moment. I think a lot is at stake. We shall see that. Now, yes, we got almost to the final slide. And that's an important thing I wanted to say to this audience. You see, We have one experiment. That is what we know. Biosphere on the Earth. If there is or was life on Mars, that's almost certainly is or was related. That's not going to be it will last for 3 days and everybody, okay, yes, I mean, they were very close, so it's related, fine. Europa might be, that's one of the Galileo moons, might be an independent experiment, we shall see. But what you are excited about, of course, is that if you look into the universe, how many habitable planets you will find. Now in the PNAS, one of the latest issues, there is an analysis, look at it, which shows that it may not be the case that we are a typical solar system at all, because this particular distribution of planets is extremely unusual according to the analysis of the exoplanets that we know. That means that sadly, despite the fact that you discover more and more exoplanets every day, it might well be the case that we are extremely unique even in the astronomical sense. That means that life is probably even more valuable than anybody thought after the Copernican revolution, where they said, we are a typical case, it's just you shouldn't put the humans in the yes, the humans not, but the habitability you can put into the center. So please take care, because I'm not sure that human civilization has a long-term future. Actually, the fact that you don't see civilizations around is probably a combination of two things: that they are rare because the conditions, as we just discussed, may be very rare, even astronomically. And even if you get to that stage through the relevant major transitions, you might actually kill yourself very nicely. Not the biosphere, but intelligent life, that's easy to kill. So good luck. Thank you very much for your attention.
Hi, thank you very much for the talk. I have one question. One of Eigen's assumptions was that the code should be digital, but as Ada Jonat posed, the three-dimensional structure of RNA is not necessarily digital. I mean, you could have several sequences that could fold into similar functional states. So how could this improve or affect the stability of the hypercycles?
Can you be more specific about the question? I don't get what you want to know.
Yeah, that the RNA is not entirely digital, as was assumed by Eigen in his hypercycle model, because of the folding.
Yeah, but that doesn't, the question is whether the inheritance is digital, and the genotype-phenotype mapping is another thing. Right? You can have, many to one mappings and this kind. That doesn't change the importance of the digital inheritance. It's just that it influences how you can climb the fitness landscape. There are many models of it. If you look at the Fontana-Schuster papers about, how frequent is a certain structure in shape space for RNA molecules and what it entails for adaptive evolution, you will find the answer to these questions, but these are fascinating but more technical. It does not contradict anything in the conceptual domain.
Okay, John.
So you won't be surprised that I'm upset that my favorite transition's been subsumed into one of my other favorite transitions. But let me have you consider the following thought experiment. So I talked about it today, and other people have, is that stem that comes from between eukaryotes and LICA, there's all kinds of stuff that happen in there. If we could order some of those events, and we may not be able to, but if let's do the thought experiment that we could, like what can happen with the origin of animals, right? So you have these actual transitions to the transitions. Would the origin of sex and meiosis or the origin of certain parts of the eukaryotic cell then be relegated or increased to a major transition? So I know why you lump them together, because we can't distinguish the order of events, but are they important enough to deem as major transitions on their own? Because probably there was an order in the end.
I believe so. I also have a favored order, but I cannot prove it at the moment. But yes, so at the moment I think that whatever the order is, they were more like the phases that Andrew Burke proposed. They were steps of one major transition, which was the origin of the eukaryotic cell. I think that's more even with the whole picture now in the revised table. So even if I understand, whether phagocytosis or mitochondrial, so I don't want to make phagocytosis one major transition, mitochondria another one, meiosis a third one, because that's, you have this proliferation of transitions, and then, like gold is precious because there is not much of it.
Whole genome sex by meiosis is pretty distinct amongst those, I would say. And it's different than bacterial sex. So you and I should have a beer tonight and talk about that.
But at the moment, this is because of these problems. This is what I feel is the balanced view of the system.
It's just a comment that I wanted to make about this transition between prokaryote, eukaryote, or an archaeon and a eukaryote. It's just provocative. At what moment it stopped being an archaeon and starts being a eukaryote? What defines one? Is it the nucleus? Is it the mitochondrion? What is it that, I don't know at what moment the transition actually occurs. If there is a moment, it's a gradual transformation. And at what moment stops being an archaeon and starts being a eukaryote or a proto-eukaryote?
That's an interesting question. I don't want to give an answer now, but I outline, what kinds of things one should have in mind when attempting an answer. Cavalier-Smith lists about 20 traits that you find in all eukaryotes, but not in the prokaryotes. So one thing would be, all these 20, and unless you have it, you haven't made the transition, right? That's one cop-out. Look, these are definitions, right? And definitions are not falsifiable. If definitions are internally consistent, they are not falsifiable.
You're right, but it's inherent in any transition, even at what moment you start being a metazoan.
Yeah, but
it's the same thing with anything.
I want to make a comment which is related to that, but from a dynamical point of view. So imagine a transition towards protocells. Unlinked genes, floating or on a surface, and then you start building up a protocell. Joel Peck asked the question, can we construct a transitometer that we put into the system and it says you have transited to 90%? Can you? Now, I think that in the dynamical sense, there can be an answer in the future, but only if you can map dynamically the major transition dynamically as a dynamical system onto something which the physicists call a phase transition. If you can apply then the conceptualizations of phase transitions, then it will help you in turn to answer what kind of transitometer you can apply. But it's at the end of the paper. Good luck, I cannot solve this problem. So,
Could we have the rest of the lights up? Yes.
Well, thank you very much. I want your opinion on the author lectures, and how do you think about this obsession to find the last common answer of everything, the last common answers of life, the last common answer of animals, the last common answer of eukaryotes? Is this okay? And if we find this, what is next?
Well, I think that's actually very unlikely that you will ever have a final answer. That's because of the nature of history. Historians know too well that history is not out there. You construct it and reconstruct it always on the basis of available evidence. Certain ideas that you infer are more stationary than others, but you know, one single new fact, new document in history, one single new miserable creature in some remote place can overthrow all these ideas. And I think that there will be some convergence, that the convergence is at best asymptotic, right? I don't think that you will ever know for sure, partly because many things have been lost and the information is cannot be recovered for many things, right? But it's important because we biologists, this is how we think about evolution, branching processes, occasional anastomosis. So I think it's really a part of our knowledge to try to sort out what has happened.
Questions from people who haven't asked questions? She's asked 100 questions.
Here.
She's got the mic. Oh, well, go ahead. Nobody pays any attention.
Maybe in the future one should shoot a mark, with a red dye, Okay, sorry, yes.
Do you think that for the case of filial transitions, we might think of fitness in a slightly different way than for the other kinds of Darwinian processes, in the sense that fitness is almost having to fulfill a very specific function?
You are absolutely right. It's more like artificial selection than natural selection, because the genetic evolution of the system as a whole has set the criteria under which these Darwinian systems are working. It's absolutely true. This is what I think. Moreover, in the case of the nervous system, I think, and we have some calculations in models that show that if it's true, it's mind-boggling. It is likely to be the case, and again, we have calculations that show it, that the evolutionary process could be much more efficient in the brain than any time before in the wild. And one of the reasons is that because of the Hebbian plasticity, you can actually modify the mutation field in such a way that is not available for free living organisms, which means that the generation of variation turns into the direction of hopeful solutions. So there is a synergy between the neurobiological learning and the evolutionary process, and algorithmically it gives you advantages. None of them has individually. If that's true, you have to take it with a good grain of salt. But if that's true, that's really an amazing finding, that evolution becomes much more efficient where you didn't expect to be at all. We shall see.
I wanted to ask you a more general question about the technique, the intellectual technique to address problems. And it's about how do you deal with these leaps from the general picture, the big picture, to the small things, and then back to the big picture, and then to the small things, because it's a process where you have to be dealing with both sides, and you seem to be very developed in that sense. So,
thank you. There is, I am working in a place in Munich, which is the Parmenides Foundation. And there are two institutes there. One is the Institute for the Conceptual Foundations of Science. The other one is the Institute for the Study of Thinking. And the concept of the optimal cognitive distance seems to play a very important role how humans think about the problem. Because in many cases, the failure of thinking is a consequence that the people are either looking too closely at a problem. You can document it, with, in concrete, or they take it, from very far, and then they miss the details. But according to experience, the most successful problem solvers are who oscillate, between the two, right? Zooming in, zooming out, and this goes, with repetitions. And moreover, it's getting complicated. The speed of the oscillation probably also matters. So, in order to do it well, so it's not a likely process. It's unlikely by itself. So you have to learn it. You have to train. You can train yourself partly to have this oscillation, and then work out for the particular problem the optimal cognitive distance or the distribution of these distances. You are absolutely right. Practice, right? Practice is really an important thing in this case. It's hard in the beginning, because it's you feel insecure, but then you become brave with time.
And maybe could you maybe you have an example of how a practice could be done?
Yeah, think about it. I mean, that's all. And think about it, and think about what I said. That try to distance yourself from the details. Try to make the more abstract version of it. Once you think that the picture is clearer, then you can return. It's a recursive thing. So, and then, but it's not specific to me or whatever. That is specific. That is also happening, in good management. A good manager, when managing a company, does exactly that. Bad managers are stuck at a single level, and then you can have extremely adverse consequences. But there are concrete studies behind that.
I think that could be the last question. But before I hand it over to Federico, who is, I think, going to make some formal concluding remarks, this is the last opportunity for all of us who are the beneficiaries of all of the amazing work and accomplishments of Juan and Berenice and all of the others who've worked on this. This is the opportunity for us to give them a very rousing round of applause, expressing our thanks to them for the amazing work that they've done. And if you jump up and down and yell, you'll get warm too. So.
I will be very brief because it's too late. But thank you for coming to Mexico and to U.N.A.M. to this symposium of revisiting the major transitions in evolution. As you can see in this, or maybe it's not very obvious, but we have a lot of help from U.N.A.M. From two major centers, of research centers, our Institute of Biotechnology and also the Center for Genomic Sciences. We got a lot of help also from the National Research Council, the Council of Science and Technology, CONACYT, and also a lot of help from our own money. I'm not kidding. On Saturday, I saw Bere's father climbing in a ladder and trying to put some dark stuff so that we can avoid the light. But that was around 8 o'clock in the night. And also in the lab, people from my lab and others have been very generous. Antonio Lascano's lab also, to provide for coffee, which you cannot get that money from anywhere. Not CONACYT, not the U.N.A.M would pay for a coffee break. So if there was one only coffee break, now you know why. So in the poster is not easy to see, but we really had a lot of help from our national university, from the main authorities which provided, our institute provided for the transportation, but also provided for the loan to pay the hotel, which the grant will be available until next probably 2 weeks. So you don't have to be arrested in the hotel. You can leave to whenever you want. So the idea, and this is, I would like to tell you, and I told you at the beginning that Iñaki Ruiz was responsible or someone to blame about this, but it's because, well, let me tell you this, because I was talking about this with Rosie when I was complaining that she was not allowing me to ask for questions. But I said, well, the students are the important ones. You are an old guy and you don't, maybe you have a good question, but let the students to have a space. And that's what she really did, and she's a wonderful job. I agree with you, Tonio. And indeed, we all wish that many of you students became imprinted to work in evolution, imprinted to work in the origin of life, imprinted to work in the word you just heard these 3 days. I hope that you commit your future work or your future studies in some of these issues, that wonderful transitions and pseudo-transitions or whatever you major ones. Not so many or not whatever if they're not major. So who organized the symposium? The passion, the enthusiasm, the talent, the joy for understanding evolution from undergrad students. Many of them are from the undergrad program in genomic sciences, which you know them because they were your chaperones. Obviously, one of them is Juan Escalona. And many others that you know because they were your personal chaperones. I'd like to tell you a few words about Juan, and I will be very brief. Juan is an excellent chef. I mean, an excellent. He spent almost 2 months in the U.N.A.M restaurant, which is in Copenhagen, #1 restaurant in the world. He was chosen, and he asked one of the letters of support or the recommendation was for me, and I have the odyssey to say that he was a bigamous because he was in love with science, and he was at the same time in love with culinary arts. And it's been very difficult for him to decide where to stay. But wherever he stays, he will be extremely successful because he's generous, he's bright, and he works like mad. So the other students are grad students like Bere, which also is. She's also extremely talented. Let me tell you a few words about her. She's a poetess. She writes poems, and she paints watercolors, beautiful watercolors, and also works like mad. So she's also, she has been for several years my course assistant, and I always had these wonderful input ideas how to improve the course, and so other graduate students from other the School of Biology are here, and also should be thanked for their enthusiasm. And who else is to blame? Well, I already told you how this idea started in Sevilla in 2013 when I heard a talk from Iñaki Ruiz. And I came back so enthusiastic, with a passion, and said we should organize one, invite him. And then we realized that this was only part of the major transitions and evolutions. And since the book had been already published 20 years ago, so it was the right time to revisit this book and this major transition. So I talked to Juan and Bere at that time, and their families are thanked here because they have been working also very generously to help Juan to prepare some surprise for you speakers a little bit later. And also Antonio Lascano, because he was always very, very enthusiastic and always prompted to give us advice whom to invite. Sometimes people could not come because they had previous commitments, so we immediately went over with him and asked him, whom would you recommend, or whom would you like to come? And he kept very close with us to organize this. The Three Musketeers and D'Artagnan. And obviously Eros, because he answered very soon that he would love to come, and he thought that this was a very good idea. So thank you, Eros. And all of you for kindly and eagerly accepted to participate. Some of you were full of work, and some also of you have health problems, job problems, whatever, but you decided to come, and this has made the difference. Also many of other colleagues from all over Mexico and the world, and U.N.A.M authorities, CONACYT, I already told you, the Morelos Council of Science and Technology, and our directors from our research centers, and Nayeli Quinto, who is, well, my political niece, is daughter of my wife, Carmen Quinto's sister, who made all these beautiful designs. She didn't ask for any money. She was, and also Geo Estrada, who is sitting there, please stand up. She had also worked a lot to her sister, her daughter, Pizza, also worked with the printing. So one thing is designing this, and the other thing is to print all this. And grad students from my lab and other colleagues, and here are the guys. Also Julian, who is a former student from the undergrad program, and now is using him with a microphone. This is Geo. Julian is now at the Max Planck Institute doing his PhD. Jesus, who is just finishing just like Juan, the undergrad program, and many others that I didn't have a chance to take a photograph. But I deeply thank you for your enthusiasm, for your commitment, for your passion to do to make this dream come true. And with that, I would like to pass the
We would like to give you a certificate of participation. Then first for the speakers. Yeah. To John Loxton, thank you very much. Thank you. Simonetta Gribaldo. Stuart West. Linda Manzanilla. No, muchas gracias. Eric Davidson, who I think is not here, but we will give him. Sharrahim Takbesh. Thank you very much. Robert Berwick. Luis Delaye. Antonio Lascano. Evelyn Fox Keller. Thank you very much. Leslie Husko. Thank you. Richard Michaud. Thank you. Bruce Alberts. Thank you. Rosie Redfield. Thank you. Thank you. Thank you. Thank you. Iñaki Ruiz. Mauro Degli Esposti. Ada Jonat, who is not here, but we will give her. Ada Jonat. Ers San Mari. Thank you. Thank you so much. And Jose Mendez, who is not here with us, but Jose Mendez. And thank you very much to all of you for being here. Thank you very much also to all the presenters who presented our speakers. Thank you very much to all of you.
There's just one other thing. I would like our host team to please stand up I don't think we would have made this without you. Thank you so much.
Well, this is it. Thank you.
Oh, one more thing before you go. I have another announcement to make. It's just a small one. At El Colegio Nacional, Carl Zimmer and Dr. Antonio Lascano will be holding a presentation on Monday the 30th. So at 5 o'clock p.m. So in case you want to go, I'm sure you will. Thank you so much.